Welcome to Science Frontiers, a space for discussions in the frontier areas of science, technology, and society. I'm your host, Binay Panda. Scientists observe, experiment, and analyze data to get to some conclusions. And when they think that the outcome of their research is ready to be communicated, they write up their results and publish their findings in scientific journals. Usually, their work is vetted by other experts in the field by a process what is known as peer review, which is managed by scientific journals. Rewards like promotions, awards, and accolades depend on scientists' publications. However, scientists, like any other human beings, also do come under pressure. Pressure to get a promotion, pressure to get awards, and to get patents that help in commercialization of their discoveries. This pressure leads to greed and often temptation to take shortcuts or engage in unethical and fraudulent activities. One way this can happen is when scientists fabricate data that go into those journal publications. When wrong information or data gets out, that leads to misinformation, which can undermine the confidence in scientists and science among general public. Unethical practices or scientific misconduct need to be checked and corrected to build trust. However, it is important to note that not all mistakes published in scientific journals are a result of misconduct or deliberate act by scientists. Many a times they are genuine oversight or human errors and are usually corrected when found. In today's episode, we shall speak about genuine mistakes and scientific misconduct by scientists leading to wrong, fake, or doctored images getting published as parts of their journal articles. The role of scientists, scientific journals, and journal editor in that process and what we can do to correct those. To discuss this, we have a special guest today, Sholto David. Sholto is a trained molecular biologist turned science integrity expert, and he is speaking to us today from his home in South Wales, United Kingdom. Sholto, welcome to the forum and thank you for joining.
Hi, Vinay. Thanks for having me.
So before we start, I want to tell our viewers that although Sholto had been doing this image forensics for a few years now, he rose to prominence and limelight by highlighting a series of articles containing wrong images published by researchers from a very high-profile cancer institute called Dana-Farber Institute, an affiliate of Harvard Medical School, that he published in a blog post called For Better Science. And that has led to the retraction of 6 papers and corrections in as many as 31 others. Sholto, before we go into some of the more substantive issues and questions, can you tell our viewers a bit about yourself, your childhood, where did you grow up, go to school, college and university, your training as a molecular biologist, and how did you become interested in doing the detective work?
Yeah, sure. So I grew up in the southwest of England, and I went to York University for my undergraduate degree, which was in biochemistry. After that, I went and worked in Northern Ireland for about a year as an analytical chemist for a veterinary pharmaceuticals company. I was there for about a year, and then I went to do my PhD at Newcastle University. So my PhD was in cell and molecular biology, and it was to do with bacterial copper storage proteins. So it was microbial cell and molecular biology. And my PhD took 4 years, and after that, I went to work in industry. So I haven't stayed in academia. I worked for a company called Oxford Biomedica. They manufacture viral vectors and vaccines and stuff like that. And my job there was analytical science. So everything to do with developing assays for quality control, qPCR, ELISA, cell-based assays, stuff like that. It was a nice job. I enjoyed it. I was there for 3 years. And then after that, I moved and I worked here in South Wales very briefly for a CRO, a contract research organization, again, in the analytical role. So ELISAs, biomarker assays. And I was only there briefly. And then last year, I decided to leave my job and do other things for a while, although I'll have to go back to work eventually. And I'm looking forward to it, to be honest. But, I've been interested in maybe fringe science for a long time, the alternative medicine and stuff like that, maybe 5, 6 years longer than that, really. It's just been an interest of mine, and I used to be interested a lot in how alternative medicine is researched and maybe how that kind of stuff ends up in scientific journals like acupuncture, herbal medicine. People research that, and sometimes their studies published in scientific journals. It's quite an interest of mine. And then gradually over the years, I've become more and more interested in just errors or mistakes or potential fraud in different kinds of science. So initially I was interested in clinical research more, how clinical trials may contain errors and systematic reviews of clinical trials might contain statistical errors. So I used to be quite interested in that. And then when I started writing comments on PubPeer, which is a website where you can write comments about research papers, I noticed that a lot of people are writing about problems with images, particularly images that have been published twice or appear twice in the same paper. And I've ended up looking at a lot of images myself for signs of Photoshop or just simple copy and paste errors. These are quite surprisingly easy to find in journals. So I've been doing that for about 3 years. Then I've been blogging under my own name only for a few months, to be honest, since the middle of last year. Yeah, I hope that's a fair introduction.
That's very good. So let's now go into some of the specific cases. And I want to tell our viewers that the cases that we shall discuss today with Sholto are not the only cases that are related to wrong images or data fabrication, and there are far too many to discuss. However, we are going to speak about the one specific to Dana-Farber Institute today because it's one of the most prestigious cancer research institutes in the world, and most, including myself, look up to the work done by the researchers there. And when things like this are highlighted, it shakes the confidence in scientists and science in general. Now, Sholto, there are 37 of the Dana-Farber papers now been either retracted, as I mentioned, 6 retracted and 31 corrected, but you have raised concerns on nearly 57 in your blog post. Why did you decide to go after the papers published by a few of the top scientists there and not somewhere else?
Yeah, so just a couple of points. So far, they've announced there will be 6 retractions and 31 corrections, although we're waiting to see what the journals do. So this is Dana-Farber's perspective. They want to retract 6 and correct 31. But we don't know if the journals will accept the corrections. They may also choose to retract, or we'll have to wait and see how that plays out when they send those to the journals. 57 is the number that Dana-Farber gave, but it's not a number that I've ever given to them. So I think in total, over the last month or so, I've ended up sending 61 papers to them. My initial spreadsheet, which I sent by email, contained 50, and then I've added a few more after that. And the blog post was more narrow. It focused on just a few, maybe 30 or so instances and examples that I was most interested in. Why did I decide to go after this institute? Well, I'm interested in kind of everything. So if I write comments on PubPeer, it will include what people might consider to be very low quality research, like paper mill type activity, commercial, almost fraud. And I comment on high profile institutions as well. And I think all scientific research is important to be criticized. From whoever writes it, wherever they come from, it all deserves to be criticized. When it comes to Dana-Farber, I was following researcher to researcher, so I started a project to go through all of the papers in one journal, and that caught a particular author in the US, and then I followed his co-authors when I found problems with the images, and eventually it led to Dana-Farber, where I found more image problems in their papers. So yeah, I have a cast a wide net, and whoever falls into it, I will write about the problems where I see them. And I think it's important to criticize all literature.
Absolutely. Now, on the Dana-Farber case, a lot what you found and published on your blog post, as you already have mentioned, are posted on the website PubPeer. A site that allows, just for our viewers to know, a site that allows others to comment anonymously on published papers or preprints or anything that is posted publicly and have a DOI or digital object identifier. Just that our viewers also know, the posts that you put on PubPeer with the name Mycospirilla arachidis, which is the scientific name of a pathogenic plant fungus. I'm not sure why you do that. Maybe that's one of your famous or very dear fungus. But you have posted more than 2,100 comments in PubPeer, including many of the papers that are published by Dana-Farber. In your interview to Nature that was published in 24th of January 2024, you said that you had noticed a specific image of mouse with bioluminescent image been superimposed on others, and you did that, or you could find that when you located the ear of the mouse in multiple images exactly at the same place. Was this the specific paper that in PubPeer that piqued your interest in the institute, or something else that actually happened between then and till you published your blog post, so that you decided to go ahead and dig more on those issues?
I can't remember the first paper that I found, which was published by Dana-Farber Research, which really made me dig into this more. Um, it, as you know, I write a lot of comments, so it all becomes a bit of a blur. Yes, you're right. I found an especially concerning paper where there was a figure which was supposed to show mice in different groups and mice on different days. So you can imagine there will be rows and columns of images of mice, and there was a mouse which appeared on multiple days and in multiple different groups. And if you look very closely at the position of the mouse's limbs, you can identify it if it is or isn't the same mouse. Now, you have to be very careful because mice all look pretty similar, but there's only so many ways you can lay out a mice, I suppose, is the way to put it. When it comes to my PubPeer username, anyone who comments anonymously is assigned a random username from the Tree of Life. So mine's a fungus. If you signed up with an anonymous account, you'll get a random username from the Tree of Life. I didn't choose it, but I don't mind it.
I see. Okay,
Yeah,
that explains the name, and that also explains. But the point I was trying to make is there one particular journal paper from Dana-Farber that actually piqued your interest, that you started digging more?
No, I'm pretty prolific. If I find a mistake or an error, I'll just keep looking. I think it was probably histology images from a particular researcher, Ruben Carrasco, and those are thin slices of tissue, right? He's published a number of papers, or a couple of papers I spotted with histology images which are being copied and pasted and rotated, and that caught my interest, and that would have been what's kind of kept me motivated to look further. But yeah, I mean, if I find one or two image errors or other types of errors, then I'll tend to look for a long time. Yeah,
Right.
the errors which stand out to me are usually the ones which involve images of animals, and maybe when you see Western blots, not just copied and pasted, but rotated or perhaps stretched or changed in a way that you may not think is natural. Because obviously, when you see an image of a Western blot which is being copied and pasted once, I think that can happen by mistake. Lots of errors like that can happen by mistake. When you start to see more kind of what I would describe as manipulation or maybe editing to Western blots, then that becomes more concerning, and it will cause me to look more carefully, I suppose. But in this case, I can't remember anything in particular. I'm very busy with these things. I don't really stop. So yeah.
Right. That explains. Now, you detected most of the image manipulation in the images, like you mentioned, in Western blots or duplication of the animal images or the cell line images, by using a tool called Image Twin, in addition to detecting many manually. For the benefit of our viewers, Image Twin is a commercial image detection software made by a company based out of Vienna, Austria. Now, Sholto, you mentioned in some of your writing that you've got free access to this software. Is it correct that you have? And can you tell our viewers how exactly does this software work?
Yeah, so I just want to make one quick point. When it comes to these image problems that I've identified, some of these, which were sent to Dana-Farber and were written in the blog post, were identified by other people, right? So some of these comments have been on PubPeer for about 10 years. Now, when I came along, I independently discovered some problems at Dana-Farber, and then I rolled them all into the blog post and the notifications that I sent to the Institute. But it's just fair to say that some of these image problems have been detected by other users on PubPeer. Elizabeth Baker, you've spoken to before, had found at least one of them, I know, and there's a couple of other people who are named commenters. So just to know that I don't want to take credit where it isn't all of my work, right? Some other people have contributed heavily there. But although I did find a lot of the new errors and then wrote the blog and everything like that. But I just wanted to make that clear. So yes, I use Image Twin. I wouldn't say that I used it to identify most of these, but I don't keep track carefully. Image Twin is very useful. It's very powerful, particularly at certain types of images. And yes, I have been very kindly given free access to the software. Sorry, can you just repeat again, what was the specific question I was supposed to answer?
Asking is how does this software work?
Yeah, okay. So it looks at the most simple level, right? It looks for similar areas within images. So if you upload a PDF to Image Twin, it will scan the PDF and it will see if any of those images look similar to each other in that PDF. And the other thing it will do is it will take those images and it will scan them against a database it has of previously published scientific figures. So it does two things, right? It looks for similar images within the same paper, and then it scans them against a really large database of previously published scientific images. So its database contains a lot of open access papers and stuff like that. Papers that are behind a paywall, Image Twin won't be able to check for you. And when it finds similar images, you can click, it will highlight them, and it will show them side by side. So you can decide whether that's a problematic duplication or an appropriate duplication, right? So in a paper, there are sometimes images which we expect should look the same. For example, the control condition for a group of cells, it might be shown twice, or maybe a Western blot with loading controls could be shown twice. So it's useful. It's very powerful. It's very sharp-eyed, I suppose, and it highlights the similar areas. And then you still have to have some kind of input yourself. You have to understand the experiment to know whether or not this is an appropriate or inappropriate duplication. And it also has, as you can imagine, like most software, there's a rate of false positive and false negative findings. So it doesn't find everything that you can find by eye. And then it quite often highlights things that look similar but aren't actually the same.
Right. I mean, so basically you said about the software being useful for both intra within the paper and inter between the papers published. However, you also mentioned that it obviously scans everything that is in open domain. That
Yeah.
means there is a possibility that what one finds by using image twin could be far less than what's out there, because unless the paper is out there in the open domain, the software would not be able to scan. Am I correct?
Yes, that's right. So papers that are paywalled or inaccessible, Image Twin doesn't have in its database. I don't think it has a complete database of open access papers either. But it's a very large database and much more powerful than you can do by eye.
I mean, they
But
say,
yes,
sorry,
so
they say it's about 50 million images on their site.
50 million images, yeah. So it's large, but it's only a fraction of the scholarly literature, right? It's not everything. And as you say, right, if it's behind a paywall, it won't be able to find that. And I think probably most historical literature is inaccessible, but it's still certainly extremely powerful.
Right. So, I mean, it's fair to say that what one can find by using the software could just be the tip of the iceberg. There could be a lot
Yes,
more
yes,
to be found.
yes.
Right? Okay, excellent. So at this point of time, it's important to tell our viewers that there are both genuine mistakes and deliberate frauds or misconduct that can cause for a wrong image to appear on a journal article. It's important to stress that if there is a wrong image in a figure, it's not always a deliberate act of fraud or misconduct on the part of a scientist. Honest mistakes do happen by everyone. In addition, science in many cases is dynamic. Many of the results may change when a different person tries to replicate the results of the old experiments, repeat it by using a different set of reagents or different sets of people. And our knowledge may change as we discover more about certain things. Therefore, I want to divide our discussions going forward into two aspects. First, the genuine mistakes, and then we'll deliberate on the misconduct part. Now, before we do that, Sholto, there are, so that we are on the same page, how do you actually define scientific misconduct? In other words, when should one categorize an image duplication as scientific misconduct and not a genuine mistake?
That's a very tough question. I think in general, we should probably be quite generous, I suppose, and accept that for the most part, it would be good to believe that people are trying their best to get it right. And there are lots of examples of images which have just been copied and pasted next to each other, and it seems like it's an error that's happened during the preparation of the figure, right? And there's a process when we take a research image, and then maybe we'll use PowerPoint or Photoshop or some kind of program to arrange figures in a panel or like in squares or whatever, and you can see that errors can happen in that process. So as you say, there's lots of examples where people might call them innocent mistakes, right? And then there are more difficult to explain transformations. For example, if you have an image of cells and then someone's used the same image of cells, but it's been rotated, that's a little harder to understand why you would choose to rotate things. And of course, there may be innocent explanations for that too. And then there are more complex where you may have an image of cells and then an image of cells which overlaps in the corner. So you can see that they're sharing an area and they've been labeled as being in two different treatment conditions. Again, so that's maybe another step up of difficult to explain from the innocent perspective. And finally, most obvious when you have, for example, someone has used a graphics editing tool to clone a particular cell multiple times in an image, right? That would be scientific misconduct. I don't think there's any innocent reason to do that. And maybe using a graphics editing tool to obscure particular areas, maybe by coloring them in black or coloring them into the background, right? Those kind of things are much more on the end of scientific misconduct. But for me, it's very difficult to draw a particularly hard line. And let's say, for example, you find a lot of copied and pasted simple duplications, and they're sort of everywhere in someone's work, then that points to a level of laziness and sloppiness that might itself be scientific misconduct, right? So I don't think you have to see these complex graphics editing transformations to be concerned about someone's work. And and sometimes you may see a very large number of simple mistakes, and that itself would point to a kind of lack of control over your work that is also a very complex question. I try not to think about it too much. Hopefully, when the errors are pointed out and blogged about, then the journals can maybe come to that decision, and people will have to investigate very carefully. But it's a tough question, and I suppose if we want to be generous, we should probably just think, try and be as kind as we can to scientists, because it is difficult, right?
Totally understand. Now, let's talk about those genuine innocent mistakes. Now, if there is one and someone detects it, whose onus it is to correct it? When should it be done? And what's the best way of correcting the scientific literature? In other words, it's pertinent to ask who bear the responsibility to find those papers that contain those wrong images to start with, and who bear the responsibility to correct those mistakes.
Yeah, it's interesting. I don't think there's anyone who really bears the responsibility to find image errors or any kind of errors in the scientific literature, right? Once we have peer review, the discussion kind of ends there. So a lot of the detection is done by amateurs or people in their spare time. So no one really has the responsibility to find the mistakes, which is interesting, right? Maybe there should be. But once one is found, I think it's generally the authors who have the responsibility to own the correction, right? Because they will have access to the raw data, and they'll be able to figure out if there is a mistake, what it is, and whether they have the raw data to replace it. And there is a process, right, for scientific papers. There are corrections. You can post the corrections to the journal, and they should be able to publish it and update your article. Or if it's a serious mistake that undermines the data itself, or something that's been quite critical, you could choose to retract that paper as well. So the processes exist. They're not especially efficient. They're not really very modern. They're still built for the kind of publication on paper style journal. It can take a long time to get these corrections through with journals, even when authors really want to make them. But they exist. So it's generally the authors have the responsibility to fix things through the corrections process, which is, it is what it is, but it does work generally if the authors try.
Right. So now that we have talked about the innocent, genuine mistakes, let's come to the 900-pound gorilla in the room, which is the misconduct and the fraudulent data and the implications. Before I do that, it's important to list out the possible types. And I have listed out the 5 types, and please feel free to add or delete any of those. And these are very broad classification. Now, let's start with the most egregious one, where all the data is fudged, entire paper is fabricated, no experiment was ever performed. Of course, this happens extremely rarely, and there is no doubt in anyone's mind that the paper should be retracted. The second case, where experiments are performed, data is presented, but in some figures, the data do not actually match with the actual data obtained in the lab. And there is a deliberate act on the part of the researcher to alter or fabricate the data. Now, in this case, where the conclusions derived using the fraudulent data or results do not hold good at all, what should one do? So that's the case 2. In the third case, the underlying data in some figures and not in all do not match with the actual data. And the main conclusion or the associated conclusion doesn't hold good once you take that data out. In the fourth case, a panel, not the entire figure, a panel in the figure, because as you know, modern papers in particularly biology, experimental biology contains multi-panel figures, that means the data in a panel is fabricated, and taking out the particular panel in a figure or replacing that data with the good data doesn't actually change the main conclusion of the paper. And the last case, where there is something wrong or data is fabricated which has become a part of the supplementary figures, not the main figure, and the main conclusion has nothing to do with removing that particular supplementary figures. Now, how should one deal with each of the situation? And I mean, there are two broad categories we should talk about. One, where removing the fraudulent image or the panel doesn't really change the conclusion of the paper. And in another, it does. So how should one deal with situations like this?
Okay, so we're now wandering a little bit into more opinion, which I'm happy to talk about, but I do want to say that I don't think of myself as much of an expert on these things, and I really love details and I love images, and that's what I like to write about. And so I feel very comfortable talking about images and pictures and mistakes. But when it comes to these bigger picture questions, I'm aware of my own kind of ignorance. So I haven't worked in publishing, and I don't really intend to. But I will answer your question just with the caveat that this is a little bit outside of my normal territory. When I think about a scientific paper, though, I think the most important things for me is the methods are described accurately, so we know exactly what someone did, and then the results shown, including all the pictures and images, are completely accurate too. So that's exactly what the experimenter received or what they got from the experiments. And as for the conclusions, actually, maybe this is a bit radical. I don't care so much for the conclusions, whether or not conclusions are true or affected. For me, the conclusions they're the author's opinion of what the results show, right? Which is maybe useful. But if the methods are accurately described and the results are exactly what was found, then you can come up with your own conclusions. And hopefully, a scientific paper, when it's well written and when you're kind of really familiar with that field, I think most people, when they're deeply in a research field, will feel like the results and the methods are the most important thing, and they'll be able to figure out what the conclusion of that results or paper may be without the conclusions ever being written. It's rather like the introduction. When you've been in a field for a long time, you probably don't spend a lot of time reading the introduction, and you may not even care so much for it. So when it comes to figures and data in a paper, which have mistakes, of course, when they're honest mistakes, then they can be corrected as long as the raw data can be sourced. When there's that evidence or suspicion of fabrication, it comes to whether you have confidence in that process, right? And when the confidence in results in a paper is being undermined, it's very difficult to kind of see the rest of the paper as being untainted, I suppose. So yeah, I have maybe a slightly more extreme view of that, and obviously some people would place a lot of weight on the conclusions. And I often see on Pub here, right, if I comment about someone's paper, they'll say the conclusions are not affected. For me, I find the conclusions to be one of the least important parts of the paper. I think methods and then results are super important, because then you can come up with your own meaning of that data, right? You come up with your own conclusions, and in the long run, I think for a lot of papers, the conclusions will always be essentially shown to be outdated or incomplete, or not wrong so much as, but when we look back in really old papers, those conclusions they won't fit in with later data, if you understand what I mean, because science is always expanding, right? So as the data in those papers, what the experimenters did and then what they found will always be true if they accurately describe it, and what they conclude may or may not be true in 50, 100 years' time. Does that make sense?
Yeah, yes, it
So
does. I just
I'm,
wanted
I'm
to
less
know.
interested in the conclusions and how to deal with individual fabrication. I suppose it's going to come to whichever editor or person deals with it. If it really is isolated, then perhaps it can be replaced. But if we're losing confidence in some figures, even in the supplementary, that then I would struggle to kind of maintain confidence in the whole paper. That's my opinion.
Now, I want to play the other side, the devil's advocate, if we may. As you know, modern science is too competitive. It involves many people in the lab, from students to postdocs to lab heads to collaborators lab, his, her, or their students, postdocs, etc., etc. It takes years for the lab heads to generate funding, years to complete the work, do the experiment, replicate your findings, and then write up the results. The heads of the labs are often very busy people. Many may have been involved or still being involved in discharging their clinical duty in addition to doing research. And it's very easy to blame them when they may not be involved themselves in what is called misconduct or fraud. And it's humanly possible to detect all the, it's not humanly possible, I meant, to detect all the mistakes in a complex paper that has been published years back. The graduate students left, there are no lab notes, postdocs left, and those papers sometimes contain, as I mentioned, 8 or 9 or 10 figures, each figures with 10 to 15 panels. So we are talking about 80 to 100 figure panels and the underlying data plus hundreds of supplementary figures and information. In the situation like this, do you really expect the lab head to know about the underlying data that has gone into each of those panels? And if that's the case, should the scientist not be legit to just withdraw or just say expression of concern to the journals? I mean, you don't certainly expect the lab head to reply to each and every anonymous post in Papier, do you?
Yeah, it's a question that people have, right? And I would say yes. I do think people should respond if they are an author on a paper. They should respond. And when we talk about lab heads at an institution like Dana-Farber, as you said, they have multiple duties. They may be in charge of very large labs, but that's a choice they make. And certainly there's this idea that scientists are under a publish or perish kind of crush or squeeze, but the rewards are very high as well. So, you know, some of the salaries these people earn are extremely high, and certainly more than I'll ever earn. And I think we have to keep that in mind, right? So they're also very well paid, very well, you know, rewarded for their tasks. So they have a great responsibility, right? They have a really large responsibility. Now, if they choose to run a very large lab, then they should take on all the responsibility with that. We all have to make choices and kind of trade-offs with what we want. So when it comes to these very, you know, senior scientists, I don't believe in blame the PhD student or blame the postdoc. I think the most senior people in any kind of organization have the greatest responsibility to get things right. So when I think about these big mega labs and these large research groups at Dana-Farber, I still think that the people in charge of those labs have the ultimate responsibility for the data in those papers. And if they feel like it's reached a situation where they're not able to review all of the data in the papers that they're named as an author on, then they should stop being an author. They should say, well, maybe it's not appropriate for me to be an author on this paper because I haven't been able to review all of the figures in it and I don't feel comfortable with all the data. And that's what I would say is people should think about taking a step back and thinking about their own responsibility and what they're capable of reviewing, rather than scattering their name across hundreds of papers which they aren't able to review, because it still benefits them, right? There's always a benefit when you're an author on a paper, but somehow people don't want the responsibility when it comes to correcting them. So, you know, I don't find it to be a strong argument, although I do understand why people make it. I think if these mega labs and these large labs, the leaders of them aren't capable of reviewing and answering questions about all of their papers, then perhaps they've been putting them, you know, spreading themselves too thinly. Maybe they should focus more on few papers but higher quality. And if the papers themselves get too long, maybe we need to think about publishing smaller papers, better quality. Just, you know, again, my opinion being from the outside of not publishing a lot of papers. That's how I'd respond to that. I hope it makes sense.
Yes, I just wanted to know how you feel. So I understand your stand on the responsibility of the lab head, but again, I'm not defending anyone, but let's get real. I mean, is it really possible or practical in a modern biological experimental science lab to actually think that the head of the lab, which controls, and I totally get it, where you see that it is their choice to have a big lab, it's their choice to do many experiments, but let's just get real. The reality is right now in most big universities around the world, and particularly the institutions which are very prestigious, the labs are big, they are running huge experiments. Sometimes the PhD students or postdocs left years back. I mean, there is no way that the head of the lab actually can know if the student, and I totally get it that you don't want to put the responsibility on the students or the postdoc said, but do you think it's really practical?
Um, I mean, honestly, yes. I honestly think that the data, when you run a lab, you should save your data in such a way that it's archived, and every university will have a process to archive data. It should be saved in a structured fashion so you can access, what experiment I did to do with this project and what day. All of that information should be available to the authors. And so if it is the case that they've made a mess when they've been archiving or saving their data, then that reflects poorly on the way they've been running their big lab. But when I was a PhD student and when I subsequently worked in industry, I saved my data and I kept lab books where appropriate, right? And these were in formal folder structures, so it wasn't a lot of hassle to go back and verify what I had or hadn't done in a particular experiment. So yeah, I think it should be possible. And if the lab leader has become so removed from the day-to-day analysis of data, that's kind of troubling, but they should be able to delegate to an appropriate person, right? So if they're themselves not able to understand a particular experiment or they're not aware of where some data is, they should have someone in the lab who's able to access and analyze the experiments that have been done to support a particular project. So I do think it's practical, and I think if it's the case that people are struggling, they should have a think about the way they're processing and analyzing and saving their data. Not to say that everyone's perfect, right? I don't want to. I'm sure there's people who might be listening, sweating a bit, like I can't access my experiments from 10 years ago. Well, I'm not saying it's perfect, but I do think it's something that is practical, should be aimed for, especially at somewhere like Dana-Farber. There's no real excuse for losing research data. Some of the stuff they're doing involves animals, it involves human samples, right? These are all stuff which should be treated with a really high level of importance when it comes to maintaining that data, right? Because you don't want to take animal data more times than is necessary. You don't want to take human samples more times than is necessary. So that data should be saved and stored in a way that it's going to be accessible for years at a time, I think.
Right. And that brings the next question that there are two different elements here that also bear responsibility, and I want to ask you about those. Number one, the reviewers of the papers. Number two are the
Yeah.
journalists. Let's start with the reviewer. Now, it's expected if you are an expert reviewer, you should have caught what you caught in an open eye in a field that you don't work. Now, somebody who's working in that field, now there's a bigger problem, I understand. Getting reviewers to do review in time, which is a thankless job, and I have spoken about it at length in my podcast before, is a bigger problem. Now, journal editors also finding it so hard to actually convince the reviewers because, as you know, reviewers are not paid, is a thankless job. Now, there certainly is a responsibility on the head of the reviewers to catch as much as possible. And then we'll talk, and what do you think about that? And then we'll talk about the responsibility of the journals itself.
Yeah, okay. So with regards to reviewers, again, as someone who's not published a lot of papers, I've published a few and I think I might have reviewed one in my life. So again, as someone quite outside of this process, my feeling is that perhaps reviewers of these papers, if you look for what you find what you're looking for, right? So if you're a reviewer of a paper, I expect they're most interested in the methods, and that the conclusions reflect what's in the results. But I suspect that most reviewers are not looking for image duplications or other types of errors that can be spotted internally in a paper. I suspect they simply aren't looking for it. So I find it, it doesn't surprise me. Now, when I used to read literature as an undergraduate and a postgraduate student, I never found any mistakes in papers because I wasn't looking for them. And now, subsequently, when I go looking for errors, then I find a lot of them. So it's to do with whether or not that's the focus. And yeah, do reviewers have a responsibility to find these things? I think so, but it's obviously difficult, and it's hard to blame people who weren't looking in the first place.
I
Yeah,
mean,
tough question.
it is because that brings us to the next second part of the question, which is what about journals? Because in many cases we pay thousands and thousands of dollars to the commercial journal, the society journals, the article processing fee for the open access journals, and they make billions and billions of dollars in profit. So don't you think that you as an individual who is not trained in the areas that you have looked for all the papers on, with the help of a software, if you could catch so many, don't you think a journal publishing house who is making tens of hundreds of billions of dollars in revenue and profit bear responsibility to employ these softwares, and many do actually, and also do some vetting to make sure if image twin can do it, a new company can do it, then why can't the big publishing house do the same thing internally? Which I'm not saying that they will catch 100% of it, but they can always employ people that in addition to using software and people's expert minds, they should be able to catch most of it. Why are they not doing it?
Yeah, so I would agree that the journals themselves have a lot to answer for here. Um, as you say, they have a lot of revenue. They take very large article processing fees, um, and they have a lot, a lot. They have a lot of the responsibility. I think if they're supposed to be responsible in some way for the quality control here, yes, the journals do have a lot of responsibility. They should be able to employ the latest tools, and with all the revenue they've had, they should have been able to develop them in-house. Um, One thing I would like to point out is there is a slight kind of unfair advantage and asymmetry here in that when I look at papers that were published, say, 10 years ago, I'm able to use Image Twin, which wouldn't be available to a reviewer at the time or even the journal. So there's a slight kind of unfairness to the fact that I'm able to go back and review papers with modern tools that weren't available at the time. Although I actually comment most of my comments are on recent papers. I don't go back into 10 years ago very often. Well, I occasionally do, but most of my comments are on recent papers. So, but yes, journals have a responsibility, and it's not like they wouldn't have been able to commit resources and develop these tools themselves. And obviously, large publishers will have access to their own paywalled papers as well, so they should be able to access lots more images than even Image Twin can. So yes, I think
Absolutely.
they have
I
a
mean,
lot of responsibility, and they've really dropped the ball. And they should also, I think, they should also be doing this work themselves in terms of going back and checking their own back catalog of papers. Right, I see that they have a responsibility for what they publish now and for what's been published previously, but they
absolutely.
don't really
And
seem to be very
this is
proactive.
exactly this is exactly what I was asking. Who bears the
Mm.
responsibility? And you mentioned that there are probably no one. I think the journals bear responsibility because they have been paid. And we are not talking about someone who is in your backyard making £1 a year. We're talking about
No,
tens of
no.
hundreds of millions and billions of pounds and dollars in revenue and profit. They are Goliaths in the industry. So if a small startup company could come up with a software and have open access, like papers in the repository, the big publishing house, certainly the commercial publishing house, or even the society publishers equally, should be able to do. In fact, they should be made mandatory that they should have a repository of all the papers, not just open access, all the papers. And for the prospective papers, do that obviously, but even going retrospective and try to scan all the papers they have published, I think the responsibility is theirs. Nobody's taking away the responsibility of the individual authors and the lab heads to bear responsibility of withdrawing the paper or correcting the paper once someone points it out. But I think the journals and the publishing house actually bear even a bigger responsibility of going back and correcting the literature for the sake of science. Would you agree?
Yeah, I mean, again, without wanting to step too far out of my feeling of where I'm comfortable with, yes, I agree that journals have a lot to answer for. They should, presumably it would be good if they would go back and check their existing literature and work a bit harder on accepting new ones. I don't want to take anything away from the Image Twin guys who've made some amazing software, and sometimes it does take innovative small startups to kind of unseat. So I don't want to take anything away from their efforts, and I don't want to assume that Elsevier could have done it better, but they've clearly had the resources at least. They could have tried.
Right.
And image twin is available to publishers now, and a lot of, as far as I know, publishers do use image twin, and there are other competing softwares available that some publishers use. I know that Proofig is out there. I know some publishers use that. So they do use it to screen articles now. So they but not all articles are screened very effectively. Some still make it through with obvious image errors. But yeah, I think publishers, since they get paid in this process, and they are, as far as I understand, supposed to have a hand in the quality control of scientific literature, they do have some responsibility here, and they just certainly don't seem to do a great job of it from my perspective.
Right. So let's move on to the next question. We all agree that genuine mistakes and innocent mistakes must not be blown out of proportion, and no one should go for witch hunts against a particular researcher. A piece in the British newspaper The Guardian on 29th of January 2024 by its science editor Ian Samples said this about you, and I quote, David stopped short of alleging wrongdoing and stressed that he did not want to create an environment where scientists felt harassed or terrified of publishing, unquote. Now, some may say your work will eventually end up helping some to harass the same scientist whose work you are flagging. What do you have to say about that?
So I want to say that my own kind of perspective on this is a little bit complicated, right? And if you read the blogs that I've written, they're pretty mean. So the blogs I've written are pretty mean. And I'm in two places, right? On the one hand, I don't want to create harassment or distress for people who have made innocent mistakes and who make a good effort at correcting them. On the other hand, I do think there should be space in science for really scathing criticisms and clear criticism of people who've made lots of mistakes. The way that I think about it, right? We have awards and we have prizes and we have ceremonies in science, right? We have very high praise for specific people. We have newspaper articles saying how great scientists. So I think if we want to allow that kind of treatment of scientists, this veneration, then we also need to probably have space for serious criticism as well, right? We can't have just the veneration and just the prizes and awards and no criticism. And so I think there has to be both ends of that spectrum if we want to have those awards and prizes and ceremonies. And the other way we could do it is not talk about the individuals, but really focus on data and papers and kind of forget the individual praise or criticism and think more about just the scientific papers. So, I have a kind of complicated relationship here. I have said, and I do believe that, it would be wrong to heavily harass people about the errors they've made. But then at the same time, I do believe that there's probably space for real criticism of scientists. And that can be like we have with politicians, right? Politicians have a lot of responsibility. They have a lot of power in certain spaces. And so we have very positive things that are said about politicians, but we also have mean cartoons, and we have the ability to mock those people in a way that we don't with the regular everyday people. And when we think about scientists at Dana-Farber, right, they get a lot of high praise. They're the top scientists curing cancer and all these kind of things. So at the same time, when they get it wrong, I think the criticism should be an equal and opposite reaction, I suppose. That's again my slightly uninformed opinion on it. And
Right.
as someone who's written
let's
quite cruelly.
Yes, and also, sometimes scientists are also human. For example, the site like Puppeteer, they can be very mean, take out vengeance wrongly, anonymously, and it's very easy. We must also add, it's very easy that one can accuse others, whether rightly or wrongly. And if the accusation happens anonymously, then the onus becomes the person who is accused to clear his or her name or their name. So that part also can actually backfire. And we have to keep in mind that while we are all in favor of having science done properly, at the end of the day, the data has to be vetted, and it has to be for public good, because we do science with the help of public money. But at the same time, we also have to draw a line that when the anonymous report comes, and there are a lot of discussions and comments and debate about whether one should sign the Puppeteers. And in fact, the danger of doing that, particularly if you are young researchers looking for promotions, grants, you know what happens. If you criticize someone, a senior person, publicly, and rightly about the paper, which should be criticized, you know what happens. It's very, your life becomes very difficult. So the case can be made that the anonymous comments are the right thing to do, but at the same time, we have to draw a line that just because somebody can comment anonymously, that doesn't necessarily mean that, one can actually go up to any end to clear his or her name. So it's a tricky issue. It's nobody knows the answer to that, but I think we got to be fair to both parties.
Yeah, PubPeer, the website we're discussing is moderated. They have moderators, and the moderators, I think they have the line about right. So they will remove criticism which is not scientific. They will remove insults. They will and they will always prioritize an author's ability to respond and have the last word. So they tend to moderate people who ask questions and write comments more heavily than they will moderate an author who wants to respond to those comments, which I think is right as well. Right, authors should definitely have the opportunity to respond. So that at the same time, I think it's important to encourage the discussion of science, even when people get it wrong. We should encourage the discussion of science. People should be encouraged to join the discussion. And we have been comfortable with anonymity in science for quite a long time in the form of peer review. We're comfortable and we understand the reason why we do anonymous peer review. So we should, I think, extend that opportunity to people to comment post-publication anonymously. But you're right. There is a risk, and we should be aware of the risk of allowing kind of legions of anonymous comments on people's papers that aren't really true or tricky or yeah. So and I would say at the moment the PubPeer moderators, they have that balance pretty much correct as far as I can tell.
Right. Now that we have come towards the end of the interview, I need to ask you the last question. You don't have a day job, Sholto, and you do this policing work completely free of cost and full time now. How do you sustain yourself? I mean, how do you manage to pay rent, buy your food, buy your beer, and travel?
Yeah, so I left my last job last summer, and I've had some savings, right? I've saved up some money. I was saving up some money. I was planning to buy a house. So now I'm spending that money. But I'm not well off, but I'm also not in a desperate financial situation. So I'm comfortable enough with the money I have. I'll stretch it as thin as I can, and I've been doing some odd jobs here and there. And recently I've had some support on Patreon, right? So I have a Patreon account, and some people have agreed to sign up and support me, which I feel quite uncomfortable about. I don't like, it's a bit, the feeling of donations just feels a bit off for me. I don't like it. But it's not a lot of money. It doesn't pay the electric bill, I'll say. Maybe just about matches my electric bill, but it's nowhere near my rent or food costs. And so I'll go back to work eventually. But this year I was thinking about going traveling or something, and I'll spend the money I was planning to spend on a house, and then I'll go back to work, and I'll be a bit more motivated to work when I've got no money in the bank, I think.
Right. No, I mean,
But
we want to really thank you for your time. And I think from the community as a whole, what you're doing to vet those papers and give a chance to the authors to correct is really hugely and immensely beneficial. I just want to tell our viewers that genuine mistakes do happen. And whenever there is a deliberate mistakes, the author should get a chance to correct. And with that, I really want to thank you, Sholto, for your time and your generous time and spending time with me. Thank you and goodbye.
thanks a lot.